Okay, welcome back everyone for the second half of the session. I hope the morning was nice for everyone and got a lot of information in there. We're going to start with session 3, which is difficult to treat infections and multi-resistant organisms. We are taking a completely different, nuanced field here. We're going to learn a lot about phage therapy and also what are the newer antibiotics coming from to use. Our first speaker is William Franks, who's going to give us an update on new and available antibiotics. I know Will from my advanced trainee years. He is an advanced clinical pharmacist with extensive experience in antimicrobial stewardship and infectious diseases, covering a broad range of specialties. Will is the lead AMS pharmacist to the Metro North Hospital and Health Service Directorates across Brisbane. He led the implementation of novel AMS programs across the hospitals and an initiative that now extends to various niche areas, including community and oral health, which is where I work with him very closely in my other role, and corrections health services. He has a special interest in therapeutic drug monitoring of antimycobacterials, antifungals, and antivirals, and also to ensure the safe and efficacious use of these medicines with a narrow therapeutic index. Please welcome Will Franks on our next topic.
Well, thank you for inviting me to speak today. All right, I've been unofficially promoted. I'm not a doctor. I didn't finish my higher research degree, but I'll take the promotion today. So I'm going to talk today, obviously, about new and available antimicrobials, but there's some caveats to that. Newish, as in they've been around at least internationally a little bit of a while, not too long, and their availability in Australia is at the very least plausible, not necessarily actually available. And so I thought, similar to Patrick, I thought gram negatives are really the area of clinical practice that we're really struggling with agents to find an efficacy for them. So I thought I'd focus on the 3 bottom agents on this slide. So cefepime and mirtazobactam, sulbactam durlobactam, and sulopenem. I confess I was a little bit dubious about including sulopenem for reasons that I'll go through a little bit later, but nonetheless, I think this slide, which is from a great article in The Lancet, it really just highlights the difficulty we are having with finding new agents that are effective for gram-negative bacteria, especially carbapenem-resistant Acinetobacter. Steno is obviously a problem. And difficult to treat drug-resistant Pseudomonas also has some problems. But at the very least, we do have some agents that are increasing armamentarium. So cefepime and mirtazobactam, if we get there. You can train a person to build a PowerPoint, not a person to use it. There we go. All right. So look, it's a fairly cefipime's been around for donkey's years. We know that. We know it and love it. It's a great drug. And mirtazobactam is a new beta-lactamase inhibitor. And it's been put together with cefepime to improve its stability against ESBLs. It retains the same issues, though, ultimately that cefepime has, in that it's not really effective against enterococci. There's not activity against anaerobes. We don't have activity against Steno, Acinetobacter, carbapenemase reducing Enterobacterales. So it does have a limited place in therapy. But nonetheless, it's a new drug and with newer activity. So mirtazobactam is the new thing on the block, and really it's just tazobactam with a hydrogen molecule attached to it, the lovely little blue there. But what that does is it does actually improve the neutrality of tazobactam, and that enables it to get into the bacteria cell just a little bit more effectively. It does seem to have improved MICs against the Enterobacterales, which is again useful. This isn't necessarily ESBLs, it's a generalized slide. I'm a pharmacist, so I thought we'd go through some pharmacokinetics. In terms of its actual dosing, it's the same as what we would use cefepime normally for. It's 2 grams, just add the half gram of mirtazobactam. But the way they've done it, and I think it shows the progression of pharmacokinetic pharmacodynamic relationship and how we use that for antimicrobials, in the sense that how that it's been trialed, they've done it over prolonged infusions. And that's actually the recommended way in terms of the product information, how it's available overseas. So 2 hours for urinary tract infections, but also that prolonged 4-hour infusion period for hospital-acquired pneumonia or for those with augmented renal function. We're building a picture of what it's like and penetrating into the various tissues, and certainly, at least in healthy people, it penetrates into the epithelial lining fluid quite reasonably. In pigs, it does penetrate into the muscular and some of the bone reasonably well as well, but it is a renally cleared drug, and so that does result in us dose adjusting it for those in renal impairment. Now, I've got a comment here, SAS Australia. That's a theoretical. And for people who don't routinely practice in Australia, what we have in Australia, we have a mechanism for drugs that aren't registered here, but have gone through the regulatory process in a country overseas that has similar regulatory requirements. We can obtain it. But we have to go through a special paperwork process and an application. It's largely tick and flick, but nonetheless, it has to be done. The inquiries I've made thus far for cefepime and mirtazobactam would suggest that we can't get our hands on it easily at this stage. And so I have no idea on pricing or lead times, but in theory, we could actually get it. It doesn't actually appear on the portal nonetheless either. So there's been one phase 3 study for ex-BLIFEP. They're creative, the boffins, aren't they? And look, it was trialed in patients, adult patients with a complicated urinary tract infection. About 50% of them, give or take, had pyelonephritis, and a small proportion of them had a bloodstream infection, roughly 10%. Now, it was trialed on the standard dose that I've put on the screen, the 2.5 grams 2 hourly because it's UTI, for 7 days. But the comparator now, look, it was piptaz, which is, we all know and love it, but it was trialed at 4.5 grams 8 hourly. And the thing with this that I've got in the comments, that the CLSI breakpoints for tazocin have since changed since the study was published. At the time of publication, the CLSI breakpoint for piptaz for resistance was 64. It's now 32. And so what these actually results in terms of efficacy to me are unclear, and have we actually done it in the right population? I apologise, the numbers for percentages of ESBL on the screen are wrong. It was actually about 20% in each arm. So even though we're trialling it in a group of an organism that we have concerns for treatment for, we haven't really trialed it potentially in the right arm, or have we potentially picked the right drug? You sort of wonder whether it was actually better challenged against meropenem. But nonetheless, it was a double-blind non-inferiority RCT, and at least under the circumstances here where they needed resolution symptomatically and microbiologically, cefepime and mirtazobactam in this particular setting did prove to be non-inferior to piperacillin and tazobactam. All right, sulbactam, delbactam. Thank you, Patrick. That was a nice explanation on it before. I have a Pac-Man slide. Look, it's called Zacduro for carbapenem-resistant Acinetobacter. And the way that it works is sulbactam acts as the beta-lactamase inhibitor, and delbactam, which is a non-beta-lactam beta-lactamase inhibitor, that effectively protects about the degradation of the sulbactam from the Acinetobacter. It's given in partnership and in imipenem. In theory, we don't need to give imipenem, but in the trials, they give imipenem because it was done in HAP VAT. And ultimately, the concern is that you've got to cover those other organisms. And so its place in therapy without imipenem is really unclear. But nonetheless, it does provide really great coverage against carbapenem-resistant isolates of Acinetobacter. And we can really see that the susceptibility profile is maintained in the presence of carbapenemase-producing isolates. And again, cefidrochol raises its ugly head again, but we can just see that difference there, how it drops off once the carbapenemase inhibition production starts. So delbactam, sulbactam, basically we have to give 2 drugs, and they're 1 gram each. Now, we can get it in Australia. When we've inquired about it, it's about a 2-week lead time from the United States, and it costs us about $50,000 per week of therapy. And that's before we add on the imipenem, which, because is now SAS for us as well, isn't as cheap as it could be as well. Now, look, it's the drugs are fairly, both of these are fairly similar in terms of their pharmacokinetics and their half-life and things like that. But the way they did it in the trials also is they gave it over the Durlo Solbactam over 3 hours, and the imipenem was only over half an hour to an hour. So I confess I don't really know what to make of that, but it's nonetheless an interesting pharmaceutical side note. Patrick's mentioned the ATTACK study, and look, as you mentioned, the mortality was good, in the sense of, for Delabactam, Sulbactam, it did actually have a benefit. And the colistin arm, the patients unsurprisingly had some acute kidney injuries. I think relevant to our transplant cohort, though, the immunosuppressed population, to my reading in this study, it's unclear. Presumably there are, you don't get a carbapenemase-resistant Acinetobacter easily, and usually there's some other things going on there, but yeah, there didn't seem to be a large hematological component or solid organ transplant component to this particular study. Sulopenem. I think sulopenem is really a case study in the difficulty in getting a drug registered. I think the good people at the pharmaceutical company, which I can't recall, it'll be on the next slide, really had a lot of faith in this drug. They've run 4 phase 3 RCTs, 2 of which have proven to be not non-inferior from the criteria set by the FDA. Its place in therapy is limited, really to those ESBLs and EMCs, but it does have oral availability. And I think an additional complicating factor, especially for our transplant patients, is its licensing is really only for uncomplicated urinary tract infections. And so I do sort of wonder about where its place in therapy in Australia in these immunosuppressed cohorts would be. But nonetheless, it does present us with an additional option. It doesn't actually have UCAST or CLSO breakpoints yet. We only have the FDA MICs to speak of. But as it currently stands, at least in Australia, the isolates that we've tested, and there was this is from a New South Wales study. Our isolates in Australia for sulopenem are not too bad. They're generally on the lower side of that MIC. I think what this particular slide also tends to highlight is there's a cheaper drug up in the top corner, which is a bit hidden, called pivmecillinam. It's been around for a number of years, and it's widely used for urinary tract infections in Europe and Britain. We do get it in Australia occasionally, but it's not part of our standard armamentarium. Now, sulopenem is co-formulated with the oral version is co-formulated with probenecid, which again I find this particularly intriguing. It's a bit of a dogma that with the beta-lactam that's renally cleared, that you don't give it with probenecid because it's going to reduce your it's going to reduce the amount of drug you get into the urine. But nonetheless, they've this, and I didn't read far enough into the development process to how they exactly landed on this. But ultimately, it does improve the systemic availability of the drug, and taking it with food is also quite useful. It's a bit of a 50-50 metabolic process. I've not inquired as to whether we can get it in Australia. Technically, we can. It would just whether we would or not is the next question. The good people at Iterum Therapeutics really thought they were onto a winner. They ran 4 trials, all phase 3, and it really does highlight the challenges we're finding in getting new good drugs to market for gram-negative bacteria. So the top one is the most recent one. This is basically a non-inferiority study versus Augmentin. And look, in this particular case, it did have it met its non-inferiority criteria. But they did explicitly have to exclude isolates that they found were in a microbiological intent to treat side of things anyway, at least, of the Pseudomonas and the Enterococci. Basically, all the organisms that are just not common in UTIs, because Pseudomonas never gets into the urine. The other study for uncomplicated UTIs was with ciprofloxacin. Again, just with young women and uncomplicated. They used a Cipro dose of 250 milligrams, 12 hours. And I mean, yes, it's 5 days versus 3 days, but I don't think that myself is unreasonable. That's pretty standard for what we do for an uncomplicated UTI with alternative beta-lactams. They spent a lot of time explaining everything else but their primary outcome in this particular paper. and really just trying to highlight the fact that, yes, we had to do all this microbiological clearance as part of our licensing for the drug. And that's where the issue shows up with sulopenem in the complicated UTI program, in the sense that they have to do a urine check for clearance microbiologically at the test of cure. And certainly where they found in the complicated UTI program and in the uncomplicated UTI, when they're comparing against Cipro, is Cipro, when it was susceptible, is better at and resulting in that clearance in the urine at test of cure. And subsequently, that's what their argument was, that because symptomatically, these patients in the top 3 studies, the results were largely equivocal in terms of their symptomatic cure, but the microbiologic cure was more dubious. In terms of what that means in terms of the greater ecology of using these antibiotics, it's not entirely clear to me. Uh, but ultimately, they've run 3 trials and they've managed to get it across for an uncomplicated UTI in women only. They also ran it for complicated intra-abdominal infections, and they used the IV version with an oral tail of sulopenem. Um, Hazarding a guess as to why they use these longer durations, I expect it's probably because they just wanted to make sure there wasn't a duration failure as opposed to a antimicrobial spectrum failure. But they certainly went for longer courses than we would advocate for currently, in terms of going up to 14 days, where we'd advocate if there was good source control for up to 5 days tops and ending it there. Now, again, the clinical response was pretty similar, but it was considered not to be non-inferior. But there was a lot more oral step downs in the sulopenem, primarily because there was a greater risk of resistance to augmentin or ciprofloxacin in the ertapenem arm. But there was a decent proportion of ESBL antibacterials. And in this particular arm, there were more serious adverse events in the sulopenem as well. There are some exciting other agents on the horizon potentially for gram-negative bacteria. Ceftobitin, which is an older drug and overseas is available for both respiratory tract infection and urinary tract infections. It's being trialled with a couple of new beta-lactamase inhibitors, so ledaborbactam and zarubabactam as well. Cefepime taniborbactam, look, it was found to be better than meropenem in its particular complicated urinary tract infection trial. As of yet, at least according to Google, the FDA has not approved it. They're still waiting on some other bits and pieces from the company, but it potentially represents another source of a decent antibiotic. Zidibactam with cefepime, that trial has been completed, but at this stage, the paper has not been published, so I haven't put any results on that one. But as Patrick mentioned before, zarubabactam, it's been trialed with a lot of antibiotics around the track, and I've just listed a few there because it has potential in vitro activity against KPCs, but also the metallo-beta-lactamases. So I thought I'd talk about phosphomatic GPICs. It's been around for a little while, at least in lower grade clinical trials, but the availability context has changed in the last 18 months in Australia. It's got relatively broad spectrum activity against yeasts and aspergillus, less so against lamentasporum and mucorales, but it does potentially have a place in those complicated mold infections. Uh, I'm going to try and get this word out, but basically it's a prodrug and it forms mana GPICs, and it inhibits the conserved fungal glycosyl phosphatidylinositol GPI anchored wall transfer protein. And intelligently, they've just abbreviated that to GWT1, and that affects cell linking of all mana proteins in the cell. And ultimately, it's about how the cell is able to adhere to surfaces. It's an orphan drug designation by the FDA. Technically, it's not available outside of clinical trials. However, the expanded access program opened about 18 months ago, and we can get access to that in Australia for patients who have struggled to be treated with a pre-existing azole, or they've had side effects and things like that. We have one patient on it here locally. In terms of its pharmacokinetics, look, it's given both orally and parenterally. And it does have some drug inhibitions, unsurprisingly, with an antifungal, and it's got a reasonably long half-life. we're really dealing with early phase trials here. So look, in the phase 2, it seemed to have some impact on both yeasts and molds. In the phase 2 mold study, there was at least one yeast in that particular trial. They were going to do a few more patients, but they ultimately decided that they think there's some money, and they've terminated that phase 2 trial study early to focus on the phase 3, which is still recruiting. The expanded access program is through WEP Pharmaceuticals, and basically you just have to sign up and sign your life away to get access to it, but it can be done. However, we've just noticed that with the geopolitical context of the world recently, that our timeframes for delivery have actually been impacted on this, which is concerning. Hopefully it won't be long-standing, but whether that's going to affect us in other ways. So this is my final slide. I think it is anyway. There is an increasing range of mostly cephalosporin combinations that are becoming available. We can get them in Australia, but it is hard to, and they are expensive. Certainly in my preparation for this presentation, I had a bit of a scan to see what was out there for Pseudomonas aeruginosa. Look, the parenteral options do potentially give us some treatment for that, but the oral options, really, the cupboard is bare, and I think that's quite distressing. At least I couldn't find much on it anyway. And my final point, I really feel we do need a mechanism for these rare, high-cost drugs in Australia. I don't think it's acceptable to say to a patient's family, "Oh, we can get this drug, but it's gonna take you 2 weeks." I really do feel we actually should have some sort of mechanism where we have them onshore, uh, when the risk is probably gonna have to be borne by government and not by the wholesalers, unfortunately, but that's where I feel about it. That's all I have. Thank you.